why we need a corpus, and specifically what is the need for biomedically annotated corpuses. So initially, what is the main advantage of an annotated corpus? The biggest advantage is the information extraction framework generation. Whenever we have some corpus, it can be used for generating information extraction framework. So this information extraction framework is used for finding out useful information from raw data. For that particular reason, we see at current times there is an exponential increase in generation of different corpuses in the area of biomedical domain. Second advantage of corpus generation is discovery and extraction of structured information from the unstructured information. So structured information is that information which is annotated by the owners of the data, and that is useful for understanding of that particular data. The third advantage of corpus annotation is to have understanding of different association of different entities inside the corpus. So different entities such as we have genes and then diseases. Similarly, we have proteins. So protein-protein interaction can be understood by the help of our annotated corpora. Why we considered biomedical data, or what is the importance of generation of biomedical annotated corpora? One common example is that we have large number of publications in the area of biomedical domain. Such as if we talk about Medline corpus, so Medline corpus, it has roughly 16 million references available, and by a guess we say that 40,000 references are being added per month. So once we say that 40,000 references are being added per month, this is a very dynamic and huge increase in the size of Medline corpus. So to handle this information, we need some corpuses which defines some rules for that addition, so that this Medline corpus, it remains compact, and then for information extraction from this Medline corpus, there should be some set of rules to make new corpuses or use this information. So here I have just put some example with numeric numbers that how many number of documents are being present in this Medline corpus. As you know that this Medline corpus, it can be captured or searched using this PubMed search engine, which is written here. So here I have just shown the PubMed growth rate for last several years, although this is a bit old now, from 1997 to 2011, and you can see that there is a rapid increase through all of these years. So what we have concluded from this figure is that roughly there are 2 journals are being added to PubMed indexer at every minute. So this 2 articles per minute, it has done an explosion in the area of biomedical searching domain. Again, need of a biomedical corpora. Here I have given an example of 2 things which are basically unstructured data. This one, basically this is unstructured data, and this is input to any information extraction or information retrieval engine. So this unstructured text, basically implicit knowledge, so which is basically in the mind of users, implicit knowledge, it is part of, it comes into the information retrieval system. Then from information retrieval system, basically it is knowing of all the data. Then from this knowing of all the data, we comes to information extraction, which is basically specific data from information retrieval data. And this information extraction based data, it is provided semantic metadata, which helps or which adds contextual and background information in information extraction. And then what happens is this semantic data, it becomes advanced IR system, so which is combination of information extraction plus semantic metadata. And then this comes to our structured content. So this structured content, what does it do? It presents information in the form of explicit knowledge. So we have moved from implicit knowledge to explicit knowledge through these phases. Similarly, this advanced information retrieval system has another phase, which is knowledge discovery. So knowledge discovery is basically a step which is achieved after IR, i.e. semantic metadata, and advanced information retrieval. And this domain knowledge is further used for other purposes. If I want to put relationship between genomes, genomes mean genes, and phenotypes, phenoms basically. In a nutshell, the figure would be something like this. At the base level we have this phenome information, and at upper level we have this genome information. Now I have put different databases and data sets related to different phenoms and then genomes. So let's start from the upper part. So when I talk about genome information, we have shown three things here, which are ensemble genome, and EGA. Now this ensemble is basically genome's database for vertebrates and also some eukaryotic species such as humans, mouse and zebra fishes. Then we have this second part, which is shown by a database or data server, which has literature and ontologies related to genome information, such as we have CIT Explorer. CIT Explorer is basically a platform by EMBL and EBI people, which are basically European Molecular Biological Lab, and this platform is freely available and accessible literature resource which provides information about literature resources and different databases of genome information. Plus we have gene ontology information, which is basically a domain to provide names of different gene sequences. Once we are done with this genome information, then we come to this nucleotide sequences, and the most famous is this EMBL bank, and they provide information about this complete nucleotide sequence. Then we have gene expression. For gene expression we have array express information and different databases. And similarly we have this proteome. Proteomes has two major things, which is UniProt and PRIDE, which provides information about different proteins. And similarly this interpro, it provides information about different protein families, motifs and domains which are part of proteins. Then we have protein structures databases, which provides information about the 3D structures, and then inhibitors and receptors which are present in the proteins for drug discovery also. Then we have protein interactions, which provides information about protein-protein interaction and their effects. Then we have some these chemical entities which are at lower level than proteins, and then we have pathways, which are pathway reactome is a database which provides information about the pathways. And finally we have biomodels for different systems. So biomodels can be for different organisms for humans, such as for lungs, for brain and cardiac type things, and etc. So what this figure is showing is that there are huge variety of databases and information available, starting from genes and ending at human phenomes. As we said that there is a huge volume of databases for different structures in biological information. Similarly, there are different applications available which are related to this biological data. What are these different applications? Such as we have this phylogenetics information. This phylogenetic information is basically our information which is related to molecular evaluation and how genes have evolved during time. Then we have genetics and population analysis. So genetics and population analysis, it can also help us in epidemic type controls. Then we have sequence analysis, structure bioinformatics, genome analysis, and so on. So on this side we have shown that okay, what are different parts which we are doing in bioinformatics. As we mentioned on the previous slide, that we have genes, then we have proteomes, then we have phenomes, pathways, cellular structures, cell types, tissues, and so on. And on the left side, these are different applications which are part of bioinformatics area, and then we have to find out some relationship between these two things.